Testing. Sorry about that little problem. We had some audio issues. Anyways, I'm going to get started now. And welcome everyone. And thank you for attending our webinar. I'm Jennifer, and I am Associate Director of the R&D team at ScienCell Research Laboratories. Today, I will speak on how to maximize your cell culture, technical tips for primary cells and cell analyses. But first off, I'd like to tell you a little bit about our company, ScienCell Research Laboratories. ScienCell was founded in 1999 with the goal of providing the research community with high-quality human primary cells. Over the years, we have significantly expanded our cell catalog and now offer over 170 different primary cell types from normal human tissue and over 100 cell types from animal tissue. We have also worked to develop tools for researchers to grow and analyze their primary cells. As you will hear in this webinar, culturing primary cells can be very challenging, so we've developed specialty media optimized to support each specific cell type. In addition, we provide cell-based assay kits and qPCR arrays to quickly and efficiently gather high-quality data from your primary cell cultures. Because we have worked with primary cells so extensively, we'd like to pass along some technical information and helpful tips, as well as introduce some of our products that may be useful in your research. The first portion of the webinar today will highlight primary cells and the advantages of using them for experiments instead of cell lines. In addition, I'll provide technical tips and optimization for primary cell culture, including the importance of using specialty medium and employing the correct substrate. Lastly, I will show a great way to analyze your primary cells using our gene expression analysis kits known as GeneQuery. Immediately following our presentation, there will be an extended question and answer session with members of the ScienCell R&D team to address any questions you have regarding today's webinar. As a courtesy, please save your questions and comments for the question and answer portion at the end of the presentation. If you find yourself with a question regarding something specific in the presentation, please note your question as well as the relevant slide number, which is located in the lower left corner of each slide so we can refer to it. Also, if you would like to further reference this presentation, it will be available for download tomorrow. At the end of the webinar, we ask that you fill out a brief 2-question survey. Okay, now that we've gotten all the housekeeping items out of the way, let's get started. Primary cells are often referred to as cell lines, when in fact primary cells and cell lines are very different from each other. Basically, cell lines are either immortalized cells or cells that are derived from tumor cells. Many researchers have chosen to work with cell lines because they are typically very proliferative, easier to transfect, are low maintenance, and more convenient. The problem is that these cells often differ genetically and phenotypically from their tissue of origin, and they show altered morphology. Many cell lines have also been misidentified, are contaminated with other cell types, or are developed from a single source. Primary cells, which are isolated directly from tissue, show normal cell morphology and maintain many of the important markers and functions seen in vivo. Primary cells, though, have a finite lifespan and a limited expansion capacity, and are more sensitive to culture conditions and density. In addition, primary cells require extra nutrients to support them in culture. Although more challenging to work with, primary cells are ultimately more representative of the in vivo environment, which means that your experimental results will be more relevant. To illustrate how primary cells can differ from cell lines, here are images highlighting the morphology differences between primary retinal pigment epithelial cells and retinal pigment epithelial cell lines. As you can see, all three of these cultures look very different morphologically. One of the cell lines even exhibits a more fibroblast-like morphology, which is shown in the middle, rather than the typical epithelial cell morphology. Because of these morphological changes, you can expect to also see many differences at the functional level. The differences between primary cells and cell lines are well documented in the existing literature. In one study, which I show here, Steiner et al. Compared primary mouse brain endothelial cells to a common mouse brain endothelial cell line known as BN5 using a variety of functional markers. First, the authors noted that BN5 morphology was more spindle-shaped than the typical primary brain endothelial cell morphology, as indicated by the red arrows. Predictably, they also found differences in the F-actin cytoskeletal architecture between the two cultures. Looking further, the authors identified changes in tight junction organization, which is shown by occludin, and T-cell extravasation, shown in the picture on the right, leading to functional differences under both inflammatory and non-inflammatory conditions. Primary cells in general have many advantages over cell lines, but they do require extra care and maintenance. We know that primary cells can be very challenging to work with, particularly if you only worked with cell lines, which are generally very forgiving. Today, I would like to share some technical tips on how to handle primary cells so that you can get the most out of your primary cell cultures. Because we have extensively worked with primary cells, we are very familiar with the common problems researchers encounter when culturing them. Here, I will discuss some common mistakes made when using primary cells and the technical tips that will correct these mistakes. The technical tips will guide you from the point of thawing a vial of primary cells and go through the subculturing stage. Along the way, I will be noting critical elements of primary cell culture. First off, when working with primary cells, it is essential to remember that they are not cell lines and must be treated very carefully. Additionally, before thawing primary cells for your experiments, it is extremely important to be familiar with the morphology of the cells you are culturing and the potential contaminating cells. The figure shown here demonstrates how human keratinocyte, endothelial cell, and fibroblast morphology differs. As you can see, these cells have very different morphologies, so it makes it fairly easy to distinguish between the various cell types. In most cases, it will not always be this obvious, so you can reference the ScienCell product page for more details of correct cell morphology. A common misconception is that primary cells are 100% pure. Remember that primary cells are isolated directly from the tissue and are rarely 100% pure. For our cells, we try to get the cells as pure as possible. The image panel shows the markers for 3 different cell types from ScienCell: neurons, endothelial cells, and astrocytes, and helps to illustrate purity. The first picture shows beta-tubulin 3 positive rat hippocampal neurons among 2 non-neuronal cells with DAPI positive nuclei, as indicated by the 2 red arrows. The second image shows an extremely pure culture of human umbilical vein endothelial cells, otherwise known as HUVECs, as seen by the ubiquitous staining of the endothelial cell marker CD31. The third image, which is of human astrocytes, is also very pure, as seen by the widespread positive GFAP staining. So as you can see, these primary cells are all very pure. However, it is worth noting that as primary cells are passaged, contaminating cells can begin to grow out. Being familiar with markers for the cell type you are working with is also very useful and will further be addressed in a subsequent technical tip. Another myth with primary cells is that they can proliferate indefinitely. We strongly encourage you to use primary cells as early as possible for experiments to prevent genetic drift. For example, some primary cells like fibroblasts can be extremely proliferative and can be passaged many times, but it may not be optimal for your experiments to use cells that have diverged so much from the original cell population. Keep in mind that the reason you have selected to work with primary cells is because you wanted cells to be most similar to the cells found in vivo. So now that we have discussed the importance of being familiar with the cell types you are working with, you are ready to thaw your cells. A common mistake that is made is to thaw the vial for an extended period of time in a water bath. Primary cells are very sensitive to the thawing process, so it is important that the vial be placed in the water bath, as shown in the picture, held and rotated gently until the contents are just thawed, and then the vial removed from the water bath promptly. Remember to spray the vial with 70% ethanol to reduce the risk of contamination in your culture, as water baths are a frequent source of bacterial or fungal contamination. The next step that most researchers do at this point after thawing a vial of cells is to centrifuge the cells in order to remove the DMSO. With primary cells, we actually do not recommend to centrifuge the cells after thawing because the centrifugation procedure is more harmful than the minimal DMSO residue. We instead recommend that you skip the centrifuge step and directly transfer the vial into a sterile hood. Primary cells, as I mentioned, are extremely sensitive after being thawed, and it is very important that cells are gently resuspended using a pipette, and cells should be handled as little as possible to prevent damaging the cells. Additionally, it is important that the primary cells are not disturbed for at least 16 hours after plating them. I know if you are anything like me, you may be very eager to look and see how your cells are doing, but this time frame is very critical for the cells and will allow the cells to recover and attach. So now, after the cells have attached and grown, it is common for primary cells to be allowed to become too confluent. Unlike cell lines, primary cells can become senescent once they have reached maximum confluency. In addition, it can promote growth of contaminating cells. It is worth noting that primary cells have a finite lifespan and become less physiologically representative with each passage. Additionally, if cells become too confluent, it may be difficult to remove the cells from the culture vessel due to cell-cell interactions, and longer periods of time in trypsin are detrimental to the cells. As an example of confluency, I show rat brain microvascular endothelial cells at 70, 90, and 100% confluent. The 100% confluent image does show you how pure your endothelial cell culture is, but it is not optimal when you are trying to passage your cells. We recommend subculturing primary cells when they are 90 to 90%, 95% confluent, depending on the cell type, and planning the timeline for your experiments appropriately. Overall, you want to remember that subculturing your primary cells will not improve the health or morphology of them. This is not to say that you shouldn't subculture your cells, unless of course it's not recommended on the product sheet, but to limit the frequency of subcultures. It is important that the cells have plenty of room to grow, but passaging the cells too early or too frequently can be detrimental. If there are too few cells, subculturing is not the best way to fix this problem. If cells are not growing well, you may want to check that you are using the recommended medium and substrate, which is something I will be talking about a little later in the webinar. Additionally, when you subculture the cells, you should ensure that the cells are seated at the recommended density as shown on the product sheet, so that they perform well. Plating too many cells will mean you have to subculture more frequently, and plating too few cells can be stressful for the primary cells. So once the cells are 90 to 95% confluent, you will need to trypsinize the cells. With primary cells, it is important that they are not over-trypsinized when passaging them. If too much trypsin is used or the cells are allowed to become too confluent, cells will clump together or come off in one sheet, as shown here in the upper image. This is something you want to avoid because it makes it very difficult to get even seating of the cells when they are clumpy, and it can lead to areas of high and low density. When removing the cells from the culture vessel, we recommend using low concentrations of trypsin and monitoring cells closely under the microscope. Cells should begin to round up individually, as depicted in the bottom panel, if the correct amount of trypsin is used. Once cells have been gently removed and collected, remember to completely neutralize cells after trypsinization, because if any trypsin remains active, it will damage your cells. We specifically recommend using a trypsin neutralization solution known as TNS, which is specially formulated for primary cells, but 10% serum or soybean trypsin inhibitor can also be used. As a note, blocking only with medium will not be sufficient to stop the action of the trypsin. As an aside, all this information of how to properly subculture your cells is indicated on each of our cell product sheets, so you don't have to worry. You just follow the protocol listed on our product sheet. Additionally, if you have any further questions, our technical support team can always help you out. So very importantly, a passage and a population doubling are not the same. A population doubling is a twofold increase in the total number of cells in a culture, while the passage number is the number of times that a cell population has been removed from the culture vessel. A passage can be highly variable depending on who is doing it or whether a split of, say, 1 to 3 or 1 to 4 is being used. For all of our primary cells, we always refer to a population doubling rather than a passage number. Primary cells should also not be used for experiments past the expected population doubling, even though cells may continue to proliferate. In the image shown below, you can see how the astrocyte marker GFAP changes in human astrocytes after multiple passages. At the beginning, after thawing the cells, close to 100% of the astrocytes express GFAP, as demonstrated in the first image. After multiple passages, however, GFAP expression begins to be lost. I think this illustrates clearly the importance of using primary cells early for experiments. Often, researchers believe that primary cells can easily be refrozen. Normally, we do not recommend refreezing the cells, as this can promote a senescent phenotype and/or cause functional changes. Freezing primary cells is really a tricky process, and when done improperly may cause substantial cell death. Lastly, it is also important to remember that growth supplements, which contain growth factors and hormones, should not be refrozen because it can reduce the activity of the components and cause precipitation. Keeping these technical tips in mind when you are culturing primary cells will promote healthy and robust cells. Ultimately, if you treat your cells carefully, they will perform better in your experiments. Another aspect important for maintaining primary cells is selecting the optimal substrate for attachment and growth. There are many types of substrates to choose from, but the two main categories are synthetic and naturally occurring. The most commonly used synthetic substrate is polylysine, which functions nonspecifically based on charge. Natural substrates, such as collagen and fibronectin, tend to be more representative of in vivo conditions and often work in a more specific way through cell membrane receptor interaction. Cells can grow very differently on the various substrates, so choosing the best substrate for your particular primary cell requires careful thought and may depend on your experimental plan. As you can see from the figure, we seeded human coronary artery endothelial cells onto poly-L-lysine and fibronectin. The cells grow and perform better on the fibronectin compared to the poly-L-lysine. As a result, we recommend growing endothelial cells on fibronectin, as the cells do prefer it. Although in this particular case, fibronectin is better. Poly-L-lysine often works great depending on the cell type. It is important to remember that the growth substrate should be evaluated for each cell type. Just as an aside, if you buy primary cells from ScienCell, we have already evaluated the cells and make our recommendation for the substrate. In addition to the surface substrate, the culture media composition is also a critical component for growing healthy cell cultures. The basic elements include salts, amino acids, vitamins, and carbohydrates for energy. Serum, like FBS, is often used to provide other important elements like lipids, hormones, trace elements, and proteins. There has been a concerted effort to move away from using serum, especially fetal bovine serum. However, reducing or eliminating serum requires adding back some of the important serum components like lipids, growth factors, and hormones to achieve good cell growth and viability. In addition to nutrients, maintaining a neutral pH in the medium is extremely important because you do not want it to be too acidic or too basic. There are two common types of buffers, bicarbonate and heaps. Bicarbonate is a weak buffer that is relatively non-toxic and must be used in conjunction with 5% CO2. Heaps is an organic buffer that is much stronger than bicarbonate, but it can be toxic at high concentrations. Other than these factors, it is important to remember that correct osmolarity, or the saltiness of the media, is critical and is an element we take into account when designing medium. There are many different types of commercial culture media that are available, including classical, nutrient, and specialty. Classical and nutrient were the first media developed to grow cells ex vivo and were crucial for early studies in cell biology. Many variations of these media have since been developed and are now used with a broader range of cell types and with great success with cell lines. Classical and nutrient media on average contain about 30 to 50 components and may require supplementation with high concentrations of serum. Specialty media is typically more complex than classical media, with 60 to 80 components on average. As the name suggests, each specialty medium is optimized for a specific cell type. Specialty media are often designed to be used with little or no serum, so the media is supplemented with growth factors, lipids, and hormones to compensate. The benefit of this approach is that adding these defined components back allows you to optimize the components and concentrations for the specific cell type. So why is it important to use specialty media with primary cells? Well, you want to consider the differences between cell lines and primary cells. As mentioned earlier, cell lines are typically very proliferative, adapted to 2D culture, and tend to be more resilient to culture conditions. Conversely, primary cells are taken from their 3D host tissue and transferred to a 2D environment. Normal primary cells are less proliferative than cell lines, have more complex nutritional requirements, and are more sensitive to the culture environment. Many independent labs have spent considerable time and money optimizing their own culture media for primary cells, usually using a classical or a nutrient medium as the basal. But at ScienCell, we have developed over 70 different specialty media designed to support many unique primary cell types, including endothelial cells, neurons, astrocytes, epithelial cells, smooth muscle cells, and microglia, to name a few. At ScienCell, our specialty media are provided as easy-to-use complete kits. All of our media kits include either low serum or no serum, depending on the cell type. Here we show our ScienCell endothelial cell complete medium kit as an example, which comes with the basal medium, the endothelial cell growth supplement, which contains hormones and growth factors specific for endothelial cells, fetal bovine serum, and penicillin streptomycin. All of the components within each kit have been formulated to work in combination. So all you have to do is add the components into the basal medium and the medium is ready to use. So here I'd like to demonstrate how choosing the right basal media and supplementation can really make a difference in your primary cell cultures. To illustrate this point, we chose a challenging cell type to culture, primary human dermal microvascular endothelial cells. We looked at their growth in DMEM F12, which is already a very supportive basal medium, and compared it to our specialty media that's optimized for endothelial cells, known as ECM. We also compared these basal media with higher serum levels at 10% versus lower serum levels at 5% in combination with our endothelial cell growth supplement. Although the initial seeding density was the same, the cells responded differently in the various mediums. By day 3, it is evident that the 5% FBS with the endothelial cell growth supplement grew more rapidly than their 10% FBS counterparts. Furthermore, if you compare the 5% FBS and ECGS condition with the DMEM-F12 or ECM, the cells look dramatically better in the endothelial cell complete medium compared to the classical with supplementation, as indicated by the green arrow. We also performed the same experiment with large vessel endothelial cells and saw a similar result. Overall, this indicates that the combination of optimized basal medium and supplementation is extremely important, and I think this really drives home the point of how specialty media can improve your primary cell cultures. While some medium works well across species, it is also important to remember that the same cell types from different organisms may have different media requirements. For example, these are primary renal proximal tubular epithelial cells from human, mouse, and rat. The left image shows human cells grown in epithelial cell media optimized for these human cells. When we try to grow the same cell type isolated from mice in either the human optimized media or the animal optimized medium, it is evident from the growth and morphology that the mouse cells prefer the animal medium. On the right, a similar result is seen with the rat cells. Their growth is much better in the media optimized for animal cells. These slides have demonstrated how choosing the right media can impact the health of your primary cells in culture. And the happier your primary cells are, the more relevant your results will be. At ScienCell, we have invested the time and energy to optimize specialty media for each primary cell type, which is more cost-effective than developing and making your own supplemented classical or nutrient medium. So here I've compiled some key elements for successful primary cell cultures. As I've noted several times, it is critical to treat your primary cells with extreme care. Also, be familiar with the cells you are working with and use cells as early as possible for experiments to prevent morphological changes from occurring. Additionally, using a specialty medium especially designed for a specific primary cell type is highly beneficial, as each cell type has unique requirements. Lastly, if you use primary cells from ScienCell, please read the cell culturing protocol on the product sheet thoroughly, because we've already optimized the culturing conditions for you, and also remember to plan your experiments well before thawing your cells. So I just talked about ways to improve your primary cell culture using specialty media and specific techniques. Once you have good quality cell cultures, you will need a way to analyze them and characterize your cells. Although at ScienCell Research Laboratories we offer a variety of products to analyze cells, today I will briefly introduce you to our gene analysis product, Gene Query qPCR array kits, which can be used to analyze your primary cells. One way to compare two different cell populations is by analyzing how they differ in their gene expression. We recommend using qPCR to do this because it has high statistical power and is flexible in its design. qPCR takes advantage of the fact that gene expression can be measured by detecting the amount of mRNA present in a cell for that gene, since gene expression is regulated by mRNA turnover. In qPCR, a small amount of cDNA is used as a template for a pair of oligonucleotides designed specifically to amplify a portion of the target gene. A fluorescent dye, such as cybergreen in the mixture, will fluoresce when bound to double-stranded DNA, and that fluorescence is measured after each cycle in a thermal cycler for real-time quantification of the PCR product. In addition, qPCR is quick, sensitive, and a lot more affordable than other high-throughput methods, and is why we selected it for our gene query arrays. Aside from basic reagent and apparatus costs being less expensive, the sample concentration requirements for qPCR are much lower due to qPCR's sensitivity. ScienCell's gene query qPCR arrays are an affordable approach to gene expression profiling with minimal requirements on your part. The assays are high throughput, targeted, and have highly reproducible yields. Our pre-designed kits use qPCR to assess genes arranged in relevant panels according to specific biological pathways, cell type and function, or disease, and based on extensive literature research. A list of highlighted kits is shown here, but we offer many more on our website in the gene query section. Notably, we have unique kits for many different cell types of primary cells, including Schwann cells, endothelial cells, epithelial cells, astrocytes, microglia, and melanocytes, just to name a few. So, in addition to saving you time otherwise spent designing primers, the gene query kits also save your resources because they only require a small amount of cDNA for fast and accurate results. As little as 0.1 nanogram of cDNA can be used per reaction to detect and quantify target gene expression. We also include 5 housekeeping genes for data normalization and 3 internal controls on every plate so you can be confident that your qPCR is working properly. Since proper primer design is critical for successful qPCR arrays, which is a topic we covered in our last webinar, ScienCell has the most strict primer design criteria to ensure quality results. All of our primers arrive to you validated, optimized, and designed under the same parameters so you can compare results from different experiments or even between labs. Our primers are optimized for use with primary cells, but they can also be used with stem cells, cell lines, and whole tissue extracts. The gene query human astrocyte cell biology kit layout is shown here to give you an idea of how these pre-designed kits might work. Columns 1 through 8 are the 88 most relevant astrocyte cell biology genes we selected. Column 12 contains the 8 controls. Examining our astrocyte cell biology kit more closely, you can get a sense of how these kits can be used according to your research needs. Main genes to assess basic cell biology aspects such as differentiation, maintenance, and signal transduction are included and can be easily grouped according to their functions. To illustrate this point, transcription factors have been highlighted in orange here. Additionally, genes often misregulated in various relevant diseases such as epilepsy, Alexander's disease, and Rett syndrome have been included to help detect disease pathology and are shown here in the light green or sea green color. In the next slide, I will illustrate how this particular kit can be used to detect differences between 2 types of primary human astrocytes that only differ in their origin, brain or retina. So here we compared human brain astrocytes to human retinal astrocytes, and in just the 2 hours it takes to run a PCR, we were able to identify 5 different genes expressed in brain astrocytes, but not detected in retinal astrocytes. From these data, we also found several genes that are upregulated in either retinal or brain astrocytes, which adds to the list of genes that can aid in differentiating between cells of the same type, but with differing origins. Using gene query to characterize your cells is just one example of how our pre-designed gene query kits can aid in your research. If you don't find a pre-designed kit that fits your needs, you can also use our website to customize an existing kit or even build your own from our database of more than 2,000 genes. Since all of our primers are optimized for the same PCR conditions, you can mix and match as you please. Ordering individual primer sets is just as easy. ScienCell conveniently offers individual gene primers in addition to the pre-designed arrays. Furthermore, their They're already organized by cell biological functions, physiological functions, or diseases. So all you have to do is navigate to the category to see all the gene primers we currently have available. You can order a few or as many of our optimized gene primers as you'd like, and if you're looking for a particular gene but can't find it, you can contact one of our gene query scientists who will work with you to get you what you need. If you find yourself lacking in genetic material for your qPCR, ScienCell also offers total RNA and cDNA isolated from normal primary cells. The versatility of gene query kits and primers makes it extremely useful in all types of research. In basic research, you can use gene query to study, identify, or compare relevant cell signaling and biological pathways in normal and genetically altered samples. Gene query can also be used to evaluate how your cells may respond to stress conditions and to validate your reporter assay or microarray data with higher resolution. In translational research, gene query can be used to assist in drug screening to identify novel target genes, genetic markers, drug action, or metabolism mechanisms and aid in drug development. In clinical research, gene query could be used to test drug safety and efficacy. Your research is very important to us at ScienCell, and I hope the technical information I've presented here today will be helpful to you. To summarize, your culture conditions heavily influence the state of your primary cells. Choosing the right growth substrate and specialty media for each of your cell cultures will help in optimizing cell growth and viability. Once you have established your culture conditions, gene query kits and primers can help you characterize your cells in a high throughput and targeted fashion. If you'd like any more information about the products used and featured in this presentation, please go to our website at sciencellonline.com. Again, if you feel like you've missed something or want to see something again, this entire webinar will be available for download tomorrow. Remember to fill out our brief 2-question survey before exiting the webinar. Tune in for our next webinar, which will provide technical tips for our gene query arrays. Thanks for listening, and we'll take any questions you may have now. You can either type your question or you can call in. And we'll be muting for just a brief second so that we can switch and one of our other scientists, another Jennifer, will be answering some questions while I take a quick break. Hey guys. I think so. I'm going to answer a few questions that were asked in the middle of the presentation while Jennifer takes a quick break. So one of the questions was, do we test our cells for mycoplasma? Well, yes, we do. We also test them for HIV-1, Hep B, Hep C, bacterial yeast, and fungi. And this applies to all of our primary cells. Another question that was asked, was how often should I change the cell culture medium on primary cells? Well, it kind of depends on the confluency of your cells and also the cells you're using. But in general, we recommend you change medium after 16 hours of plating them just to remove residual DMSO and dead cells. And thereafter, every other day, every 2 to 3 days. And you can kind of get a good sense of when your media should be changed based on the media color, because spent medium usually changes color. The pH tends to change for it.
Okay, so another question was, can I use my own medium or other commercially available media for culturing science cells' primary cells? Our cells have been cultured and tested in in our own medium, which we recommend for you to use. They've been adapted to our specific formula. Using other media may be fine, but it may also yield unsatisfactory growth due to the adaptation process. So we don't I mean, it may not end horribly, but we don't really recommend that. So one question was, why is centrifugation more dangerous than DMSO for the cells? It's um, it's just really rough on the cells. And the DMSO, it's not great if you leave them in there for too long, but the physical impact of centrifuging they're kind of groggy when you first wake them up from being frozen. So they're susceptible to shear forces and mechanical impact, and centrifuging them can squash them basically, or it kills them. Whereas the DMSO, they're kind of used to it, so they'll deal with it for the 16 hours or so that it takes for them to settle and for you to change the media. Um, do we have specific tips for transfection of primary cells? That depends on what cells you're using. I mean, it's different for different cells because some cells are more sensitive to transfection than other cells. For instance, endothelial cells are kind of well known for not being fantastic at being transfected or infected, but epithelial cells, I think they're a little bit hardier, so they can take it more. But as a general answer, um, we we have transfection reagents that are specific to certain cell types. If you go to our website, you can see the details for those, and hopefully that that helps. If you have a more specific question about transfecting primary cells, you can ask it and try to address it. Um, let's see. So how does our media compare to other specialty media on the market? We have a lot of specialty media because we carry a lot of primary cell types. Our media is designed to be easy to use, so the grow supplement that comes uh, with each media, it comes in one tube, so it's easier to put it all together. Other companies tend to provide the different components of the supplement individually, which can be time-consuming to assemble and leaves more opportunity for pipetting error or contamination. Our media is also very competitively priced compared to other specialty media on the market. Oh, someone raised a hand. I guess I'll, I'll,
Hello.
yeah,
How
go ahead.
damaging the addition of penicillin, streptomycin, any antibiotics can be to the morphology or the genetic of primary cells?
Um, I'm sorry, can you ask that again?
How damaging addition of antibiotics like penicillin and streptomycin can be to the morphology of primary cells?
Oh, um, well, in yeah, let me give this back to Jennifer. Hold on. Hi, this is Jennifer. Um, so in general, you can add penicillin streptomycin to primary cells, and that's not really going to affect them. Sometimes, depending on what experiments you're doing, I know people working with stem cells may avoid adding penicillin streptomycin because it can cause some changes to cells. But we test all of our cells also with penicillin streptomycin, and the cells will do fine and survive in this situation.
Thank you.
welcome. So if we don't have any more questions, that will end our webinar today. Thanks so much for attending.